You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends.
Share the ability to spontaneously pick up DNA from outside themselves. Called exogenous DNA molecules, and deliver them to oocytes at fertilization. So we'll talk more about that because that sounds very interesting. So Corrado, thank you for coming.
Thank you for inviting me.
So what got you into studying, I guess maybe conception or sperm and egg dynamics?
Well, it's a quite old story. It started many years ago when I've always been interested in fertilization, the combination of the two gametes that generate a new life. Mm-hmm. And I spent many years studying the chromatin, the nuclear organization of the sperm head. And at that time, I was using a very popular technique that was to prepare nuclei for sperm and then expose this nuclei to a nuclease that cleaves the DNA. And this would cleave the exposed DNA, but would preserve the DNA that is complex with protein, nuclear protein. So that could give a map, an organization map, a map of how the chromatin in the sperm head is organized. And
can you repeat that a little bit? So you did something to the sperm to reveal how the DNA is organized in the sperm head? Or was there more detail than that?
To reveal the chromatin organization, how the DNA is organized in the sperm head. You know that the sperm is a very, very small cell, and the DNA is very compact inside this. So one way to characterize how the DNA is organized with the protein inside the sperm is to isolate the nuclei, the sperm nuclei. So from the head sperm, that the sperm is made by a head and the long tail, and the nuclei is all the DNA. It's content in the nuclei of the sperm head. So we isolate this nuclei and expose this nuclei to a nuclease. The nuclease is an enzyme that cleaves the DNA, and it cleaves the DNA when the DNA is exposed. But if the DNA is complex with protein, then the nuclease would not be able to cleave the DNA. So we can discriminate between the exposed part of the DNA and the protected part of it. And this could give us a kind of map how the DNA is organized in the sperm. Is this clear?
Yeah. Is it organized very differently than in our normal somatic cells? What did you notice?
Well, we didn't know that at that time. So that's what we wanted to know. Exactly what we wanted to. It is very differently organized because it's very packed. It's a very small nuclei compared to the nuclei of normal cells. So the DNA is much more packed in the sperm head than in normal. So
what are the relative sizes of the? It's half the material, right? But what size is the nucleus of the sperm compared to a normal cell?
The DNA is the same amount. It's exactly, but it's much more packed. It's about It's about 50 times smaller. It's very smaller. It's much smaller than the size of the normal. So then We extract the DNA after the digestion of this nuclease, and we analyze this DNA in an electrophoretic analysis. We run in an electrophoretic gel to see how the DNA has been fragmented. And this was the method to which we wanted to learn more about the chromatin organization. And once we just occasionally we found that if you expose the swimming sperm to the nuclease, then the sperm immediately die in a few seconds. And when we extract the DNA from this sperm head, we saw that the DNA has been degraded. So, in other words, you don't need to prepare the nuclei and to expose the nuclei to the nuclease. Well, you simply have the suspension with the swimming living sperm. You add the nuclease, and the nuclease will penetrate in the sperm head and cleave the DNA. And so that was a very unexpected result. It was not done on purpose. It was just observed occasionally, but it was a very significant, because it means that the sperm, although it's highly packed, has an access which connects the inside of the sperm with the external environment. Is this clear?
So, okay.
Is it clear?
So what does this tell you about the sperm, that it can react to outside stimuli? It can change how it packs the DNA? What does this tell you?
That there is a connection between the external environment and then the inside of the sperm head, which is not normal. I mean, it's not expected, because sperm is considered as a piece of rock, not as a permeable structure. So when we saw that, we thought that if a nuclease can penetrate spontaneously in a sperm, also a piece of DNA could do the same. And we test that, and we found that, in fact, that the exogenous DNA can be taken up by the sperm and internalized in the sperm head. So in this sense, we thought that then the spermatozoa can be used as vectors, not only of their own genomic DNA, but also of exogenous DNA. So that was a really turning point, because a whole new field was open. So we start to, we were working with mice. So we preparing sperm from mice. We incubated the sperm with the DNA in a test tube, and then we fertilize in vitro. We fertilize in oocytes, and then we look if this the DNA that was incubated with the spermatozoa was delivered to the oocyte or to the embryos. And the answer was yes. We found that this DNA that was incubated with the sperm in vitro was delivered to the embryo through the fertilization. So the newly formed embryos contain the DNA that was originally incubated.
How did you? What does the membrane of the sperm look like? How is it different from a normal cell membrane? Have you characterized it?
No, we did not. I mean, that's, this is very well known. I'm not an expert in membrane, but the sperm has a membrane, but obviously it has some holes that allow the exogenous molecule to get in spontaneously without any special treatment.
How did you know that what was the DNA that was exogenous to the sperm? Was it tagged? How did you know that it was going to, how would you know it would show up later?
We use all different type of tests for that. We use labeled DNA, radioactive labeled DNA, fluorescently labeled DNA, and then it was specific piece of DNA. We use a plasmid, so we knew the sequence of this DNA, and then we isolated both the sperm and the embryos, and we made section of this cells, and then hybridized with a complementary probe, just to see if the complementary probe would recognize the sequence, the complementary sequences. And we found the sequences inside the sperm and inside the embryos. So there was no doubt that this sequences were taken up, internalized in the sperm, and delivered from the sperm to the embryo. So then we grow the animals from this from these embryos. And then when the animal was born, what usually it's done, you cut the terminal piece of the tail of the animal, extract the DNA, and look, you found again the original DNA that was incubated with the sperm. And in fact, we found it. And so these animals receive the sequences, these exogenous sequences, that were maintained throughout the embryonic development and were finally inherited in the tissue of the born animals. Now,
quick question: where do you think in the journey of the sperm that it has a chance to pick up this DNA? Is it while the sperm is swimming through the mother to the oocyte? Maybe the oocyte itself gives off DNA and the sperm pick it up. I don't know.
The oocyte doesn't see the DNA. The DNA, it's the exogenous DNA. It's only incubated with the sperm in a separate tube. After this incubation.
No, but in real life, where I know how you did it, but where does it? Where could it happen in real life?
This is a totally different aspect. Yes, the only I will come to this. What we found that that one implication of this finding is that if the sperm is so accessible, that's it's very dangerous because it could take up any kind of sequence from the environment and deliver it at fertilization to the oocyte, and this could compromise the genetic identity of the growing embryo. But this does not happen because when the sperm is ejaculated, the seminal fluid is contain a kind of plug that block this holes. It is a glycoprotein that stick to the sperm surface and prevent the interaction with the exogenous. So this looks as nature is aware of the fact that exposed sperm could be dangerous. So when the sperm is in the epididymous, in the body of the male, it's in the safer side and it's all right. But when the sperm is ejaculated, it's exposed in the environment, or in the case of mammals, in the female genital tract, where it could take up, as you said, could take up foreign sequences. But this does not happen because the seminal fluid that contains this inhibitory factor prevent the interaction of it. So the only possibility for the sperm to take up an exogenous molecule is when the sperm is inside the epididymous, the male epididymous. And this is what really happened in transgenerational inheritance, because as you know, the tissue that are exposed to stressing conditions, they release extracellular vesicles that contain all different kinds of information, DNA, RNA, proteins, and most of this information are stimulated in response to specific stressing conditions, like to be any physical stress or environmental stress or pathologies. And this information in the form of RNA are packed in this extracellular vesicle, like exosomes. They are released from the tissue. They are released in the circulating blood, and are reach the epididymous, and they are taken up by the sperm. And then the sperm deliver all this packed information to the next generation through the fertilization. So that's the only.
But how long are the sperm resident in the epididymis? I would think not long. So
not
that
long. No. But there is a continuous, regeneration of embryos of sperm. I think sperm, it's something like 20 or 30 days, the life of the sperm in the epididymis, but it's continues to reproduce. So there is a continuous new production of spermatozoa, so they are always there. So when if there is a wave of extracellular vesicle containing somatic information reach the epididymis, there are always sperm able to take up this information. And if involved in a fertilization process, they have the potential to deliver this to the the embryos. We have shown that, in a quite simple way. We have inoculated cancer, human cancer cell, melanoma human cell that express reporter gene, a GFP, green fluorescent protein. So this produce an RNA that it's coding from this protein. That protein becomes green, and you can see it's cell green. And so the idea is we inoculated this cancer cell into subcutaneously in a mice, and this is a cancer cell that deliver a lot of this extracellular vesicle containing the RNA, including this FP coding RNA. This extracellular vesicle are released from the cancer throughout the body of the animal. Could this reach the epididymous? And in this case, we could use the HFP as a tracer to see if the epididymous has really received this information. So we inoculated this cancer cell. We waited 45 days when the tumor was visible, you can touch it. Then we sacrificed the animal, and we isolated the sperm from this animal, and we looked for the RNA of this. And we found that this RNA was contained in the sperm, the EGFP RNA, reported gene RNA, that is not produced in the body of the mice, was present in the sperm. So that means that the extracellular vesicle were released from the tumor, were released in the circulating blood. They cross the Weissman barrier. This is this metaphoric barrier that separate the somatic context of a body to the germ line. So they crossed this barrier, they reached the epididymous, and they were taken up by the epididymous. And then
okay.
Okay, go ahead.
Why couldn't I imagine that the seminal fluid, the ejaculate that accompanies the sperm, would probably be a temporary shield. Once they're inside the female, though, I would think that would be stripped away, and the sperm would be exposed to potentially taking in DNA once they're inside the female, once they've been ejaculated.
Yes.
Did you look for any evidence that it could happen there?
Well, no, we didn't look, but since this, it's not the fluid per se that has a protective effect. It's a glycoprotein. It's a component of it. It is binding. It's sticking to the substrate where the DNA is supposed to bind. And when this place it's occupied by this inhibitory factor, just sticking there. It's sticking on the surface of this, so it prevents anyway the binding of the DNA, even if the fluid, if the non-solid part of the seminal fluid is lost. Because you are right. I mean, when the sperm is going through the female genital tract, of course the fluid is lost, but not this molecule, this inhibitory molecule. It's binding on the surface of it. So this is a.
So you've observed that. Have you ever tried to have sperm that have survived most of the passage through the genital tract or all the way to the oocyte, and then sampled them to see if the glycoprotein is still binding to them?
No, we didn't do that. No, we fractionate the seminal fluid, and we found we identified this protein. But the clear implication is that this protein it's sticking on the surface of the sperm as soon as the sperm is ejaculated. The sperm and the seminal fluid come together immediately when with ejaculation. So immediately, as soon as the sperm come out from the epididymous, would be mixed with the seminal fluid and would take up this inhibitor. So it's protected throughout the way through the female genital.
Is it a permanent binding between the glycoprotein and the sperm head, or maybe there are certain chemicals that would undo it and unglue it? I'm just thinking, and this is pure speculation.
Yeah.
What if the oocyte puts out exosomes or other material that strip the glycoproteins off the sperm, but only in the presence of the oocyte? Maybe it creates its own microenvironment that affects the sperm that have gotten to it. Who knows how, but I'm asking if you know anything about that.
Oh, I don't know anything. I mean, we have the evidence that when sperm are, if you take the epididymous spermatozoa fully permeable, you incubate them with increasing amount of all this glycoprotein. Permeability is totally lost, is deleted. You can incubate then the sperm with a large amount of any concentration, but no DNA, foreign DNA, it's impossible. So,
but is there anything that would strip away the glycoprotein from the sperm head?
Yes.
Is there anything identified that would do this? What would it be?
You can compete out this molecule, but this is done in artificial condition. It's an experiment. You can, for instance, compete the binding of this factor with heparin, which is a very strong charged molecule. But this again, you don't find this large amount of heparin in the images. I don't even know if you find trace of it. But this is again, it's something that we have done in the lab in artificial condition. In artificial condition, you can remove this binding, you can antagonize them.
Okay.
But I mean, it's
so why do you think that the sperm go through a time of vulnerability in the epididymous? What would be the purpose of leaving them exposed for a period of time? Why do you think that happens?
The purpose is exactly to take up the information that are coming from this from the stress from the soma. It's exactly what this experiment with the cancer cell inoculated in the animal tells us. So it's sperm. It's collect not only the genomic information that are contained in the chromosome, but contain also another subtle kind of information, what we call the epigenetic, in the form of RNA or in the form of variation of the chromatin, alteration of the marks, the epigenetic marks, like a methylation, for instance. And this is it's coming from the epigenetic, from the somatic. So if the soma is exposed to some stressing condition, it releases this kind of information. This kind of information reach the spermatozoa, they are taken up by the spermatozoa, and they are delivered, in my personal opinion, on purpose, because it contributes to adapt the newly generated embryos, and then the child that will be generated from this embryo. It borns already adapted to the condition that he will find in the birth, the environment. So if the parents are exposed to some special condition which stimulate or exposed to stress some organs or some part of the body, this information will be transmitted in this way on an RNA-based information to the sperm, and the sperm will deliver further to the embryo. So the embryo will already have an information that would, in principle, again, this is it's hypothetical. It's a possibility. It's not proven. This would cause the adaptation of the embryo already to the condition that you will find in the environment of birth. So it has, in my view, this has a specific evolutionary reason, adaptation.
But since sperm don't live very long, why would they need to, again, go through this period of time where they're exposed? I mean, it doesn't seem like they would need an update of DNA. Why would they want to? Why would the organism want to possibly jeopardize, unless it could improve the survivability of the sperm? Why would it want to expose them for such a short period of time and possibly expose them to negative type, changes to their DNA?
I mean, the time of survival of the sperm is not relevant because you have a continuous production. So you always have a set of sperm in the epididymous ready to get this extra chromosomal, always. Then this information would, would prepare the embryos, the newly formed embryos, to the condition that you will find in the after birth, in the environment where you will. So it's you already organize the newly formed body to be exposed to the stressing condition found. I don't know if this is clear with them. So it's in our view, it's a pure evolutionary reason why this happens. So there is this window in the epididymous where the sperm is permeable. Permeable to what? To the information that are coming from the environment. So he accepts information from the environment and delivers this information to the newly born child. So the newly born child not only have the basic genomic information, but he also has, or she also has, an additional extra chromosomal information that prepare the embryo to the environmental birth.
So what do you guess would be? I mean, what information would be useful for the father to incorporate into the sperm at this point?
Well,
why not incorporate it earlier? Why have a period where why have this window? It doesn't really make sense.
Because it depends on the environment. It's not a pre-organized situation. If the parents are exposed to some special stressing condition, then this will be also inherited by the children. That's all. If this doesn't happen, if the parents are not exposed to anything, then this delivery probably will not happen. But it entirely depends from the environment, from the style, the living style of the parents or the father. So there are also very bad consequences of that. Now it's known that there are many diseases that can be inherited by way. I mean, not really inherited in the genetic sense, but predispose the children to some disease, like a lot of metabolic disease, like diabetes or obesity, or also hypertension. That depends very much from the type of style of the life of the father. Or could reflect environmental condition, pollution, or in the contrast could also reflect the good things, like good food, a healthy life, or whatever. But that means that the lifestyle of the parent, it continues, is reflected in the life of the progeny. And this is not only for the first.
Perhaps a good practice would be, if you want to have children and you're a guy, is at least 30 days before you want to start trying, to make sure you don't smoke and you eat well and you do the right thing so that your kids aren't messed up.
Well, I don't know if it is exactly in this way, because I don't know if you can cancel in the cell the effect that you produce for years of drinking or smoking or whatever. So probably it's a cumulative effect. So I don't think that 30 days would be enough to cancel that. You know very well that if smokers, heavy smokers, need years to get them into go back to probably they will never go back to the original condition even if they stop smoking. But for many years, it doesn't really make a difference whether you smoke or not. I think after
it'd probably be a good start, at least, if not ideal.
I think one should have, in general, should avoid the non-healthy condition or healthy way, not only for your own, but for especially you reproduce yourself. I mean, that's now it's clear that this is a responsibility of the parents. And what I'm saying, all this data that I'm telling you now, that it's not my reporting literature. Our findings just the sperm behavior. But all this genetic involvement, the transgeneration involvement, inheritance of this disease and the lifestyle and personal behavior, it's how they are available in the literature.
So what does all this tell you? What can we do about the fact that sperm can take in, exogenous DNA? What does that tell us to do, or what's next in your research?
Well, no, we have. I have to say we are not really involved in the medical aspect of. We focus essentially. Our interest was focused evolutionary implications, because we think that if it's information that which the epididymous is diverge enough from the normal genetic background of the newly formed or the new embryo, it could introduce a lot of new information which are not the one that usually have the embryo, and could generate novelties, application, the relevance. And again, I'm extrapolating in a hypothetical model. If this RNA, this wave of RNA that delivered to the embryos at the fertilization is strong enough, it could generate the divergent, an ontogenic divergence could change the developing trajectory of the embryo and generate something. So we think that we hypothesize the formation of new species could come in this way. If the environmental conditions are strong, and if this stressing conditions are very strong, and therefore the amount and the quality and the nature of this RNA that reached the epididymous, it's very significant, it's very different, then it could, in principle, have the power to redirect the development. Of course,
a big question I forgot to ask you is, have you sequenced the DNA of a sperm before and after it was exposed to the DNA you gave it? And was the underlying sequence different after the exposure, or was it an epigenetic change?
I'm afraid I didn't understand the question well.
Well, you said that you exposed sperm to a specific amount of DNA, and you saw that was incorporated when when the children were born, in mice. So did the sperm take in the DNA and change its underlying structure? Did it just take in the DNA as an extra set of coding genes? Did it experience instead an epigenetic change? How was the sperm changed?
It was a pure epigenetic. This DNA that was taken out is not integrated in the sperm genome, is not integrated in the embryonic genome, and finally is not integrated in the adult. It remains as an episomal structure. So that means as a self-replicating, non-integrated structure. It's a special part. It's a different component. It's in the nucleus. It's contained in the rest of the genome, but it's not an integral part of it. And it's transmitted.
Did it remain that way in the offspring? Did you sequence the offspring?
Well, yes, we did it from the founders, from the F0 generation to the F1, and it remained as an episomal. And you can simply, you don't even need to do sequencing. You can just run a blot, extract the DNA, and look at the conformation of this special sequencing using, as I said, the special probe. So you detect specifically that specific, and it's very typical because if you analyze the tail of the animal at different times, so you cut off a piece soon after birth, you cut off another piece after 2 or 3 weeks, or after 1 month, or after 2 months, and you ask the question: is this DNA changing during the life of the animal? And the answer is yes, it's changing. And this would not happen if the DNA would be stably integrated in the genome. Once that the sequence is integrated forever. But if the sequence is not integrated, remain as an extra chromosomal or episomal, then it varies. And in fact, it's varying. And sometimes it's deleted, it disappears. Sometimes they're more stable. Sometimes after 1 generation or 2 generations or 3 generations are deleted. And that's not clear why. Sometimes it's more stable and sometimes it's not stable. But that's a factor. That's really, it's a confirmation that these sequences are not integrated.
So this means that in people, there could be these little parts of DNA that are not part of our main DNA sequence in the nucleus of our somatic cells?
Yes. There are a lot of, it's known that there are a lot of circulating DNA. For instance, the mother that have delivered children, they have the piece of DNA of the children in their body circulating in the blood. It's nothing pathologist, it's nothing bad, it's normal. But there are pieces of non-integrated in our body, which does not necessarily reflect in bad or in pathology. It's just there.
What does the non-integrated DNA do? Does it get transcribed? Does it
yes.
change the phenotype of the organism?
Yes, they do change the phenotype, and they are transcribed. We have done this using 2 reporter genes, beta galactose and one is staining blue, and the other one is staining green fluorescence. And we both found that the animals, the F1 generation and the F2 generation, the children and the grandchildren, contain these sequences, and some tissue. That's one point. It's not an homogeneous. So there are some selective tissues and preferential tissues. Reported gene are expressed, and some others are not. And it's not clear why this happened, but it's a mosaic. So some tissue are permissive; they accept this exogenous sequence and allow the sequence to be. Some other tissue do not. So, but definitely we found tissue of adult animals.
So what kind of functionality has this non-integrated DNA been shown to have? Can you tell?
Well, they look like a kind of biologically active retrogene, although the word is not correct, retrogene. So that they behave like an informational piece of DNA or RNA, produce a trait, a real trait, detected, but it's not integrated. So that in a way, in biotechnological terms, one could think it's a good system because you could introduce a new gene or a healthy gene, but without perturbing the genome, without avoiding the risk of integrating a piece of exogenous DNA into the host gene. The host gene is always a risk. You never know where this is going. Probably now with the advanced technology and specifically localized, but that was years ago. And
there's a lot of big implications for this. Very interesting. Oh, excuse me, 1 second. Is this Arnold?
Yes, this is Arnold.
Arnold, you're a few minutes early. Can you come back in 5 minutes? I'm just finishing.
Yes.
5 minutes. Can you come back?
Yes. Okay.
Please hang up and come back in 5 minutes.
Yep. Okay.
Thank you. Sorry, Corrado. We'll edit that out.
Yeah, that's fine.
Okay. So, all right. Well, we had an exogenous caller come in. So there's a lot of. It sounds like there are tremendous implications based on what you found. What do you hope to figure out in the near future?
Well, the epigenetic inheritance. Now it's a recent development of genetics, and it's really changing our belief and our thought about biology and in general about genetics. And this goes together with the fact that I think that biology is entering in a very exciting historical, which is characterized by an increasing flow of novel information. And, we learn a lot about the incredible complexity of the networks that modulate and coordinate amazingly the genome function. The even more complex networks that connect the neurons generating the brain function. And again, as I talked tonight, it's a greatly expanded concept of genetics. And in general, I think that this new fact cannot be constrained, not even explained in a narrow perspective, a casual or deterministic order as we inherited from. So I think it is necessary conceptually that opens some novel landscape perspective to be investigated differently. And something similar to what happened to physics is the advent of the quantum physics before that. What concerned my...
Well, go ahead.
Go ahead.
Well, very good, Corrado. We're out of time, but what is the best way for people to get in touch or to ask questions or read your papers?
Well, all my papers are on the Medline, or they can address a mail to me and then send. Papers are.
Okay. Well, very good. Well, Corrado, thank you for coming on the podcast. I appreciate it.
Thank you very much for the. Bye.